Hello, guys, and welcome to Muscle Histology with me, Brianna May. So, first, we're going to learn about learning objectives. By the end of this, you'll be able to identify the cellular structures of skeletal muscle cells and describe the function of each structure. You'll be able to draw a sarcomere and label all major proteins and regions. Relate the cellular structure of a skeletal muscle, including the sarcomere, to the sliding filament theory and describe the change that occurred to a sarcomere during contraction versus relaxation. Our vocabulary words. The first vocabulary word we have is sarcolemma, which is the cell membrane of muscle fiber that conducts action potentials. We then have the sarcoplasma. The cytoplasma of a muscle cell contains organelles and stores glycogen and myoglobin. We have the sarcoplasmic reticulum, a specialized smooth endoplasmic reticulum that stores and releases calcium during contraction. T-tubules, invaginations of sarcolemma that carry the action potential deep into the muscle fiber. We have the myofibril, long cylindrical organelles made of sarcomeres, the contractional units. They give the muscles its striated appearance. Myofilament, the protein filaments within sarcomeres, which can be broken up into categories. We have thick myosin and thin actin, tropin, and triaminosin. Let's give background before we get into this. What is a muscle tissue? A muscle tissue are muscle cells, which are also called muscle fibers, that can contract to produce movement. There are three different types of muscle tissues. The first one is cardiac muscle tissues, which are made up of muscle cells that are located in the heart, which is an involuntary muscle control, and its main function is to pump blood. Its fibers each have 1 to 2 nuclei and are physically and electrically connected. The second we have is smooth muscle cells, which are located in the wall of our organs like our stomach or our blood vessels, that is an involuntary control that help move substances through the body. Its uniform non-striated appearance with a single nucleus. Lastly is the skeletal muscle cells, which is attached to our bones and are voluntary so we can control them, which helps move our body parts. It's multinucleated structures that compose the skeletal muscles. All three muscle tissues can exhibit a quality called excitability, which causes their plasma membrane to change their electrical state from polarized to depolarized and send an electrical wave known as an action potential along the entire membrane. Muscles begin the process of contracting or shortening when a protein called actin is pulled by a protein called myosin, which occurs in a striated muscle, which are the skeletal and cardiac tissue muscles. A muscle can return to its original length when relaxed due to elasticity. A muscle tissue also has the quality of extensibility, and muscle tissues pull on its attachment points and shortens with force. Alright everyone, enough with the background. Let's start looking at the basic structure of a skeletal muscle. These fibers are really long cylindric cells, and they actually have multiple nuclei. That's because they're formed by the fusion of several cells during development. For starters, we have the sarcolemma, which is the muscle cell's membrane. It's like the skin of the fiber, and it helps transmit the electrical signal that tells the muscles to contract. Inside the cell, we have the sarcoplasm, which is the cytoplasm but is packed with myofibrils, which are long protein bundles that do the contracting. Surrounding those myofibrils is the sarcoplasmic reticulum, or SR, which stores the calcium. When calcium is released, that's what triggers contraction. Then we have T-tubules that carry the signal into the deep cell in the triad, which is one T-tubule with two ports of the SR. All these structures are arranged to make sure when your brain sends a signal, your entire muscle fibers respond in perfect coordination. Skeletal muscle cells also have a mitochondria, which is organelles that produce ATP, and it has a nuclei. Next, let's dive into the sarcomere, the functional unit of a muscle fiber. The sarcomere is the functional unit of a muscle fiber. It's bundled within the mitofibril that runs the entire length of muscle fibers and attaches to the sarcolemma at its end. And when the myofibril contracts, the entire muscle cells contract. So basically, imagine a disc, right? Imagine a little segment between two Z discs. There's one that completes the sarcomere. In the middle, we got a thick filament, right, made of myosins. Those are the motor proteins. Running alongside of them are the thin filaments made of actin, which the myosin grabs and pulls. Between these, we have labeled regions. The A band, which covers the full length of the myosin. The I band is where there is only actin, that part shortens during contraction. The H zone is the bare part of the myosin in the center. It disappears when the muscles contract. And the M line holds the thick filaments in place. The proteins trophin and tropin-myosin regulate when myosin can bind to actin, kind of like switch controlled by calcium. And then as you can see, I also put in here when a muscle contracts, the Z disc moves closer together. For the M line, it helps keep the myosin filaments aligned during contractions. The A band does not change length during contractions. The I band only contains thin filaments. It also gets smaller during contractions. Actin slides towards. In the H zone disappears when the muscle fully contracts because of overlapping. So I know you're thinking, how does all of this structure actually cause movement? That's where the sliding filament theory comes in. When your brain sends a signal through a motor neuron, that signal reaches the sarcolemma, travels through the T-tubules, and reaches the sacroplasmic reticulum. The SR releases calcium ions into the sarcoplasm, and calcium binds to tropin. This moves tropin-myosin out of the way, exposing binding sites on actin. Then, myosin heads bind to actin and pull. That's the power stroke. Each stroke uses ATP to release and reattach again. So imagine rowing a boat and you're releasing and reattaching again, releasing and reattaching again. As myosins keep pulling, the thin filaments slide inwards towards the M line. That's why it's called the sliding filament theory. The filaments don't shorten, they slide past one another, shortening the sarcomere as a whole. And as you can see, all of the structures are labeled, and then I gave you guys a role in the sliding filament theory, so how it connects. Now let's compare what happens to the sarcomere when a muscle contracts versus when it relaxes. In a relaxed sarcomere, the thin and thick filaments overlap just a little. The I band and H zone are both wide. But during contraction, calcium floods in, myosin grab actin, and the filaments slide towards the M line. The Z disc moves closer together, the I band gets narrower, and the H zone completely disappears. Notice that the A band, the length of the myosin, never changes. When a muscle relaxes, calcium is pumped back into the SR, Myosin lets go of the actin, and the filaments slide back apart. So, contraction shortens the sarcomere and relaxation restores it. The filaments themselves never change lengths, they just overlap. And if you guys needed me to say it again, Z-disc, relaxation, far apart, contraction, move closer together. A-band, same width, whether it's relaxation or contraction. I-band, when it's relaxation, it's wide. When it's contracting, it narrows or it disappears. H-zone, relaxation, visible. Contraction, it disappears. And then the M-line is the center of the sarcomere in relaxation, and then when it's contraction, it stays in the same place. So to wrap all of it up, skeletal muscles are designed for designed for precision and power. Every layer from the circolemma and T-tubules down to the sarcomere works together to translate an electrical signal into motion. Calcium and ATP are key players. Calcium tells the filaments when to move, and ATP provides the energy to make them move. The sliding filament theory shows how these microscopic changes, filaments sliding past one another, results in something as big as lifting a weight or walking upstairs. That's the beauty and the muscle histology. Structure and function are perfectly aligned. The end.